Thank you, everyone, for coming. My talk is going to be covering all bases, and I hope that by the time I'm done, you will know why I called it that. As Gordon mentioned this morning, Houston is a special place for us at Nanopore. For me personally, I spent a couple of months here during my PhD, but also around 7 years ago, 7 or 8 years ago, I spent a lot of time coming here visiting NASA and Sarah and sending the Mina into space with them, and it was really cool. So we're very happy to be here. We have lots of collaborations with the Texas Medical Center, and it's just really great to be here. As Sarah mentioned, I run the applications team at Oxford Nanopore. We do a lot of different things, and we're in lots of different places. When something new and cool comes out from R&D, it's our responsibility to benchmark how well that works, to find the perfect application to do it with, and to help customers like you get the best experience out of the products. So I'm going to talk about a few of these things today. There are 3 people who run the sub-teams of the applications team, Philip, Scott, and Phil, and they're all here today. So if you have any questions on anything that I talk about, please reach out to one of them or me, and we're happy to answer any questions. My talk today is going to be fairly technical, with lots of IGV plots for those of you who like those. But I do have one slide here in the beginning, just to say that everything that I'm talking about today was done on one platform. We can call all the variants that we need to on this one platform. We can sequence any read length, and really, we can cover all the bases, really, is what I'm going to talk about. The work today is mostly about whole human genome sequencing, mostly on the PromethION. I would normally use this occasion to talk a lot about our benchmarking and how accurate we are, how well we call the variants, but I decided today to more talk about how we actually see these different variations. So that is all enabled by having the reference, the human genome reference. And as you know, about 20 years ago, we got the first version of the human reference. We knew at the time that there were some parts that were missing. I guess we didn't always know how important those parts were. And then, about a year and a half ago, the T2T consortium added around 200 megabases of more reference, or more data, more sequence to the reference. And it was quite clear that a lot of the stuff that we'd been missing was repetitive and was really hard to access. I just want to have a small pitch for later in the technical update today. We will show Nanopore's only assembly of T2T assemblies, so stay tuned for that. But also, it's very important to say that the T2T consortium showed that a lot of the sequence that was missing was repetitive, but also that in the 90% of the genome that we had is actually a lot of variants that are not accessible to short reads. So some of that is called the dark genome. And my job today is to tell you that Nanopore sequencing can actually access the parts of the dark genome. But don't just take my word for it. Many of our customers have seen the same thing. So there are variants that are inaccessible to short reads because they're in repetitive regions. There are, of course, other variants that are just very difficult to access because they're really long or cover a huge region, and Nanopore sequencing can access those. I'm going to use this kind of zoom bar to indicate where I'm at in the talk. So I'll go from a very zoomed-in version looking at SNVs and indels. I'll go to structural variation, CNV, and then to larger whole genome variation and looking at how the genome interacts in the 3-dimensional space. Of course, with all of that, you get modifications as well. The last bit of information I just want to give is that many of these genes that are in the dark genome are involved in what we call pharmacogenetics. So basically, if a bunch of individuals get the same drug or get the same treatment, a bunch of them will have the desired response, but a few will have no response at all, and a few will have very serious side effects. The genes responsible for how we metabolize these drugs are called pharmacogenetics. Many of them are in really difficult regions, and so sorting out or finding out what variants people have is very important. These genes have normally been analyzed with SNP arrays and with short-read sequencing, and so we know a lot about the individual variant bases, the SNVs, but how they fit into the grander scheme of things can be difficult to assess with short reads. But it is very important to know. So let me dig into the first example. So here we're looking at one of these pharmacogenes called Sito D6. it sits right next to CYP2B7, which is a pseudogene. So here we're looking at short reads from the 1000 Genome Project. And what you can see with short reads is that there's this area of reads that have really low mappability. So with these reads sit in a region where you can't really tell if they belong to the read itself or to the gene itself or to the pseudogene right next to it. And so they're this faded out in the IGV plot. If you look at the same region with nanopore reads, you'll see that we cover this region completely. We have no problem mapping the reads because they're long enough that you can sequence, or you can just see through the repetitive region. Now what you'll see here is that there is a SNP. It might be a little bit hard to see, but that SNP is an informative SNP. If you squint, you can actually see that it is in the short reads as well. However, the 1000 Genome Project variant calling pipeline will filter this SNP out very early on because it's in this region of low mappability. So what that means for these pharmacogenes is that when you call the star allele, with nanopore sequencing and from the reference, the GetRM reference, you get a star 1, star 6 allele. But with short read sequencing, you get a star 1, star 9, which is wrong. Important thing to note here is that, as you can see, the reads are phased into haplotypes. And that's an important feature that is made easier by nanopore sequencing. So let me try to explain how that works. So here we see a bit of the genome. It's a sort of a cartoon of it. It's around 50 kb. As you know, the genome has a maternal and a paternal copy, so 2 different haplotypes. With short reads, you can easily detect the variant, but the variant sits within a single read, or a read sits within, there's one variant in a read, or there's none or one variant within a read. That means that you can detect the variant, but when you try to see which variants fit together on different haplotypes, you get a collapsed assembly, you get an unphased assembly. So you know the variants, but you don't know how they fit together on the genome. Now, with longer reads, because of just the fact that they're longer, you'll often have 2 or more variants on each read. And you can use that information, that overlapping information, to sort out how they're phased, or the phases of the genome, and how one haplotype fits with another. And that can be used to determine whether or not haplotypes, if you have effects on both haplotypes or on the same one. So as an example, here we're looking at the CFTR gene, and this is a short review of that. And you'll see that there is clearly 2 different variants here. They're both pathogenic, and they both cause the gene to be expressed, a wrong gene to be expressed, and it to be non-functional. But in this short review, you can't tell if both haplotypes are affected. It's basically impossible to tell if they're on the same haplotype or on the different ones. With the same view, but with long-read sequencing, you can see that we are able to phase based on both the pathogenic SNPs and the non-pathogenic SNPs, and we're able to phase the 2 haplotypes out. And you can easily see that here we can tell that the pathogenic variants are on different haplotypes. So this is a compound heterozygote, and both haplotypes are affected, and this is a cystic fibrosis gene, so that's not a good case for this patient or this cell line sample. And if you're thinking, can I only phase based on SNPs? Then no. We've been working with Rice University and Baylor to generate a tool called methPhaser, which Dr. Fu talked about earlier in the bioinformatics breakout. So basically using methylation to also phase the reads in places where the SNPs are the same, basically. And catch Dr. Fu's talk online after this, if you haven't already. Okay, moving on to structural variation. Moving on to repeat expansion. So here we're looking at the expansion of CTG repeats in the FMR1 gene, which causes Fragile X. In this particular case, this is a female, so there's 2 X chromosomes. And what you can see is that there is an expansion on 1. So this is a 2500 base expansion, meaning that short reads would have a very hard time counting how big that was and potentially also phasing it, because the non-expanded variant is also maybe around 100, 150 bases. Okay, so here you might think, well, this person is only mildly affected because they have expansion on 1 haplotype. However, if you look at methylation, which always comes with an nanopore sequencing, you'll see that both of these haplotypes are highly methylated. So that leads to lower level of expression, and actually this individual is affected by Fragile X. Okay. Then, we're moving on to structural variation. Here we're looking at a region that's the alpha globin genes, and you'll see that there is two genes that are very similar. This is the HBA1 and HBA2. There are segmental duplications. So structural variation can be, especially the deletions like this, can be not difficult to detect. However, they often hide within these very repetitive regions, like segmental duplications, and can then therefore be very difficult to detect with short reads. Here, however, we do see this deletion very easily, and we can phase based on both the deletion itself, but also on the other SNPs that are in there. And this person is then a carrier of thalassemia. Not all structural variation is this trivial to do with nanopore sequencing, but we can still do them. So here I'm looking at one of the most notoriously difficult genes to sequence from the dark genome. This is SMA. So SMA is a disease, or spinal muscular atrophy is a disease that can affect young kids. There is actually a gene editing treatment now, but in order to use that treatment, you need to know what the region looks like and what the sequence is of the region and how many copy numbers you have. The region itself is made up by two very, very homologous genes, SMN1 and SMN2. The disease is often caused by loss of MNN1, SMN1, and then gain of copies of SMN2. And the more you have of SMN2, the worse it usually is. So here we looked at a cell line sample derived from a patient that has SMA, and we'll see that there's 5 copies on here. So the first 2 copies are SMN1, and then you see the 2 copies of SMN2, and that SNP in red here is the one that is the one that tells you that this is SMN2 and not SMN1. And then there is a deletion. So actually, it's hard to say if the 5th copy here is SMN1 or SMN2 because the SNP that tells them apart is deleted. But because the surrounding SNPs look a lot like the SMN2 gene, that's probably it. So in this case, there is 5 copies of SMN. If you think that we've cherry-picked the best example, I will just say that we did sequence 12 different samples of complicated SMA, and we're able to call the right copy number for all of them. This region is around 100 kb, and it is fairly easy to solve this if you have reads that are longer than the region that you're looking at, the repetitive region. But you don't have to have 100 kb regions to sort out 100 kb reads to sort this out. The 30 to 50 kb reads can also sort out this region. We're also generating a tool that can help you call the copy numbers of these complicated genes. The next gene that I'm going to talk about is also one of the pharmacogenes, is Sito D6. So here we've sequenced a sample that has one haplotype that's the reference type, and then one that has some structural variation and some copy number variation. With short reads, you would see some coverage differences here, and you would probably be able to infer that you had more than one copy, but you wouldn't be able to get the base space and breakpoint information that we can do with long reads. So here we can see that there is a functioning haplotype and a non-functioning haplotype that has Sito D7, which is the pseudogene. Then it has a fusion gene of Sito D6 and Sito D7, and then it has Sito D6 that has some variants as well. And this is all spanning more than 100 kb, and so you really need the long reads to be able to sort it out. I'm moving now out to look at more of the whole genome level, in this case methylation. So here we wanted to find some interesting methylation signals. So we basically sequenced 12 tumor normal pairs, so 24 samples. There are 4 different cancer types, and we called methylation on all of them. Then we did differential methylation calling and found this region on 3 of the samples, all 3 of the tumor samples that are colon. We didn't see this signal in the normal colon samples or in the other cancer types. And so this is hypermethylation around the DAB2IP promoter. Because we did this unbiased without any assumptions about what we would find, we didn't really know what that meant. But after reading some literature on it, we found that this gene is actually plays a key role in the colorectal cancer. Now, normally with cancers, or often with cancers, you can have a lot of different levels of duplications and deletions. So we also did some CNV analysis on these samples. And what you'll see is that they're boring. There really isn't any interesting things happening at the level of copy numbers. So having the methylation information is potentially important for these types of samples. So this is sort of showing that it's very important to have both the genetic and the epigenetic information when you're looking at samples. Another place that we heard about earlier this morning that it's important to have the methylation and the whole genome is looking at cell-free DNA. So as you probably all know, both healthy and unhealthy cells will shed DNA into the bloodstream. And because we don't do any methylation, or we don't do any PCR or bisulfite treatment, and because we can sequence the methylation as it is, when we sequence these really short DNA molecules, we can look at the methylation and profile how that looks compared to the rest of the person and the organs. So what you can do is you can take that methylation information and then try to reveal what organ that tissue came from, or that DNA came from. And so without having to do a tissue biopsy, you can infer back to where the unhealthy cells are. Again, because we don't have any size selection or PCR bias, we can look at the length of those fragments. Unhealthy cells tend to produce one fragment length, and healthy cells tend to produce another. So without having to do anything other than taking a blood sample, we can infer information from both the read length of the cell-free DNA and from the methylation. I wanted to mention this whole, this next level out, the most zoomed out version, looking at how the DNA folds within, or the structure of the DNA within the nucleus. So we know that DNA folds in non-randomly to bring control elements close to the genes that they're targeting. This has been studied with Hi-C, which is a pairwise interaction, but we also studied it with an assay that we developed called POR-C. So here we are looking at multi-way interactions instead of pairwise interactions. The prep is fairly similar to, as you know from Hi-C, we do in situ digestion, then we do proximity ligation, and we reverse the cross-linking. But here's where the comparison ends. We don't do any fragmentation. So however many concatenamers you have that are fitting together is how many you sequence. And so you can then get away with these, you get out these long concatenamers where you can say, well, if A interacts with B and B interacts with C, then A and C also interact, and so on. And you get these multimeric interaction profiles. Again, because of the long reads and the way we sequence, we can look at expansions and repetitive regions, and we can look at methylation as well. So here we've looked at, again, the CGG expansion in Fragile X syndrome and how it disrupts the chromatin structure on the FMR1 gene. The last thing I wanted to end with is kind of going back to the T-to-T assemblies. So if you're interested in telomeres and subtelomeric regions, you can do a T-to-T assembly, and you can count the telomeres and look at the telomeres that way. But we've also generated a new sequencing method called Tilosseek that takes a custom adapter and enriches for the telomeric sequences. So you get the telomere sequence, the subtelomeric sequence, and the methylation, and you can count the length of the telomeres. So we just released that assay last week, and our collaborator from the Salk Institute, Toby, is going to talk right after this talk about how they've used Tilosseek to look at the telomere dynamics of aging in cancer. And with that, I hope I've convinced you that nanopore sequencing covers all the bases, and that you can really sequence from telomere to telomere and call the variants that you need because of the features of the nanopore sequencing, having long reads and methylation calling. Thank you.